All right, let's get started. So this will actually officially be the last lecture we will have this for this quarter. And then the final presentation, because we want to give you more time, so it will be scheduled, I think, on the Friday of June the 14th. It will be on Zoom. I think in the announcement, it says it will be from 9:30. I think it's at 9:30 a.m. to noon, right? So I think that's the plan, 2.5 hours. And so we hope to give everybody 5 minutes to briefly present your result. And I hope that everybody will be ready by then. Yeah. So then for the next week, what I plan to do is to use the Monday lecture time as an office hour. I will just do it online. So if you have any questions about your final project, you can join the Zoom and then ask your questions. That's the plan. And so therefore for today, I hope to cover the last step of our analysis, which is also involved in your final project, which is the finding differential express features and assigning cell types identity to clusters. Even though you have heard about the tutorial from Jiten, I will give you more insights about what's inside, what's the method inside. So before we move on to the last step, this is what we did last time, the nonlinear dimension reduction for visualization. Just a very quick recap, since it has been 1 week. We talk about, in Surat, it's the UMAP. UMAP is now the default way to go. Previously, it was t-SNE. I guess the main reason why UMAP is now used is main reason is that it's faster. And also people think UMAP has better preservation of the global topology, the relative locations of the cell groups or cell clusters. However, UMAP still has its parameters to tune. In particular, n neighbors and min dist, these are the two parameters that will determine what this figure look like. So for your final project data, if you play with the two parameters, you will see that they will look different. Okay, so that's the Surat. And then we began this introduction to nonlinear dimension reduction from these two papers published in Science in 2000, ISOMAP, which uses the geodesic distance as the input into MDS. So what it aims to do is to preserve the geodesic distance in the 2D instead of preserving the Euclidean distance in the 2D, so that the manifold structure can be shown. And then on the same issue of science, there is this LLE algorithm, local linear embedding. So this algorithm takes a very different approach. It tried to preserve a local neighborhood in the Euclidean space as a linear relationship. So basically try to learn some linear weights of neighbors to a data point in the high dimension, assuming that in this neighborhood, we can approximate it by a surface, a flat surface. Then in the low dimension, it uses the same weights, fixing the weights, but to learn the low dimensional embeddings of these data points. So it's a two-step procedure. First, learn the weights in the high dimensional space. Second, learn YI, the low dimension embedding, by fixing the weights. Okay, so they have some relative advantages and disadvantages, but both of them will rely on K nearest neighbor in the high dimensional space. That's the same thing. But one focuses more on the global structure, one focuses more on the local data structure. And SNE, t-SNE, and UMAP are the more recent methods. So SNE is the first one. Then t-SNE modifies it to make the low dimensional embedding show clearer clusters. So it will try to pull closer points in the high dimensional space closer to each other in the low dimensional space and push the far away data points in the high dimensional space farther away in the low dimensional space to exaggerate the clustering pattern. That's the intuition behind t-SNE. But what they both try to do is to preserve the so-called high dimensional probabilities of one data point given the other. right? Try to preserve the same probability in the low dimensional space. And t-SNE has this asymmetrical probabilities, that is j given I is not the same as I given j. In t-SNE, this has been changed to symmetric. By taking the average, it's been changed into Pij, symmetric about I and j. And for the low dimensional probability, in SNE, it uses low dimensional Gaussian density to define the probability. But in t-SNE, the density in the low dimensional space between y and yj is changed to t distribution. That's where the name came from. This is the t distribution density. Because compared to standard Gaussian distribution, the t distribution has the density with a heavier tail. So you have a smaller density at zero, the center, but you have heavier tails as you go away from zero. So therefore, the t distribution will have this effect to pull data points that are already closer to each other and push data points that are already farther away from each other. That's just an exaggerating effect. And UMAP has several changes. For example, it works on similarities instead of distances when it calculates this thing. And it doesn't have the normalization to make up the probabilities that sum up to one. It's just called high dimensional similarities. And this rho I here is the min dist parameter, which defines the similarity, basically. And the low dimensional similarity, you can see that it has a similar form to the t distribution. Like here, it has some subtle changes, but looks similar overall. And it has this symmetrization, which t-SNE does. It also used in UMAP. And the perplexity parameter in t-SNE, which basically defines the neighborhood size. The larger the perplexity, the fewer clusters you will have. This is also borrowed in UMAP. Okay, so a key difference between UMAP and t-SNE is the loss function. It uses cross entropy instead of the KL divergence in both SNE and t-SNE. And the cross entropy has this advantage that if you look at the shape, cross entropy means that when P is close to zero, when you find the Q to mimic or to be close to P, Q will be close to zero. When P is close to one, Q is close to one. You have this nice property. But if you use KL divergence, you can do well when P is close to one, but you cannot do well when P is close to zero. So that's why cross entropy is better as a loss function. Okay, so compared to the previous methods, ISOMAP and LLE, t-SNE and UMAP have this computational efficiency. So what it means here is that how the computational time scales with the number of data points. And in our case, number of cells. When it's n squared, it's faster than n cubic. N cubic is really slow. And then for some acceleration algorithm, you can reduce n squared to n log n, which is faster than n squared. So when you compare algorithms' computational efficiency, it's usually in the order of the data points, or people call sample size, or if you have the dimensions changing, like in your original input space, whether you input 20 PCs or you input 2,000 genes, the speed will be different. But here we're just fixing the input dimension to be unchanged. So that's why the input dimension is not here. Otherwise, you should have the input dimension, if you call it P, it should be involved in the computational time as well. So last time I ended up by talking about a method my group developed for optimizing or choosing the hyperparameters of t-SNE and UMAP. I think we basically stop here. So the idea is that we want to compare two sets of neighbors for each cell or each data point. One is based on the neighbors in the 2D embedding space by t-SNE or UMAP. One is based on the pre-embedding space. So if you use PC, it will be the 20 PC space. So we want to measure the consistency or agreement between the two sets of neighbors to indicate that, oh, t-SNE or UMAP did a good job preserving those mid-range neighbors. By mid-range neighbors, we actually look at half of all cells, because t-SNE and UMAP are known to preserve the local neighbors pretty well. The question is how well does it do for preserving the mid-range neighbors? That's why we consider half of all cells as the mid-range neighbors. So this is one cell as a good example to show that its two sets of neighbors are perfectly consistent. And for this cell, we want to show that this is the pre-embedding neighbors. This is a 2D embedding neighbors. We want to show that the neighbors change somehow. And the key here is to decide on a threshold on what we define as a reliability score, which is the consistency measure between the two sets of neighbors. So to find a null distribution, which is a concept we will visit again for the differential express feature part today. So we want to examine what will be the reliability score distribution if the two sets are random. They have nothing to do with each other. So what we did here is that for each cell, we randomly permute its gene expression values. We did that independently for all cells. Then as a result, these permuted cells should have no resemblance to each other, or their relationships to each other should be purely due to randomness. So in this case, if we apply t-SNE or UMAP to the set of permuted cells, so basically we just permute the data for once. For every cell, we randomly permute its genes. Then the neighboring relationship or the two sets of neighbors should not be matched, right? We shouldn't expect consistency between the two. So therefore, using the definition of the reliability score, we can calculate one score per permuted cell, and pooling the reliability scores together across all permuted cells, we get this null distribution. From this, we can say, okay, for each real cell in the original data, each real cell's reliability score is a high or is a low. We just compare these scores to the null distribution. And we call the upper tail given the null distribution as reliable or trustworthy, and we call the lower tail as dubious. So what we can do with this is that once given each hyperparameter setting, we run as it did, we can detect the number of embeddings that we regard as dubious. We count that number. Then if we try multiple settings, we can pick the setting that leads to the smallest number of dubious embeddings. That's how we do the optimization. Okay, so this is one example showing that in this original science paper of this species, Hydra polyp, it has the single cell RNA-seq data for the first time. And in the original paper, we see that these three clusters have this kind of positioning. And with our optimization, the positioning changed dramatically. So the orange cells become much farther away from the blue and red cells, while the blue and red cells are unified in one cluster down here. So you can see that on t-SNE, the parameters, basically whether you use the original parameters or you use the optimized parameters, it will show a dramatic change. And this figure was originally in our SAD paper, and it was recently reported by this Nature Method by a journalist who wrote an article about how do we use t-SNE and UMAP. So basically it says that dimension reduction helps to visualize high-dimensional data sets. These tools should be used thoughtfully with tuned parameters. Sometimes these methods take a second thought. The reason of this article is that there has been some recent online discussion about how t-SNE and UMAP might bias your findings. So therefore, I encourage you to read this article because it's not just our perspective, it's also the journalist also interviewed several other people who work on the topic. So that's the dimension reduction part I want to finish as the end of our last lecture. Do we have questions before we continue with today's lecture on this new topic, differential expression analysis? So we are now in the last step. Suppose that we already have the cell clusters and we can visualize them in 2SNE or UMAP or t-SNE in 2D. That's where we are. Finally, what we want to do is to find some markers or marker genes for each cluster. Hopefully, we can use those markers to assign every cluster on the cell type label based on our knowledge. So again, let's first look at what Sura does. This is the Sura's last step. Find marker genes using differential expression analysis, short as DE analysis. By default, it identifies positive and negative markers of a single cluster. So basically, you want to compare every cluster to all other cells. This is the default in Sura, but we can discuss whether this is something optimal or not later. So this ident1 is identity one is what you specify as your cluster of interest. Down here in this function called find markers or find all markers automatic. So basically in find all markers, it automatically does this for all clusters. But you can use find markers to find the markers for a specific cell cluster. So in this case, we specify cluster 2 as our interest. Now we're comparing the cells in cluster 2 to the rest of the cells by pulling the other clusters together. Okay, so let's skip this second part because it's about the fast version, the package used for speeding up. And basically, once you run the find markers, you will get the output as called cluster 2 markers because you specify ident1 as 2. So you are interested in cluster 2. And the output is a data frame in R, which looks like this. So using the head function, you are saying, oh, I'll just need the top 5 markers output. So here they are listed. And what's given out here are the p-values, average log2 fold change, PCT1, PCT2, p-value adjustment. So we will go through the results in a later slide. Let's first look at the find markers function first. So here we only specify ident1 as the class to define markers for. But there's also ident2 if you want to specify the second group you want to compare with group 1. If you don't specify it, as we do here, then ident2, the default is set to null, meaning that you use all the other cells for comparison. But let's say if you specify ident2 to be 3, then you are basically comparing cluster 2 to cluster 3 instead of cluster 2 to all cells. And there are something used as default parameters in find markers because you don't specify them and they're used. First of all, log fold change threshold, 0.1. So this means that when you look for the marker genes, you only look at the genes whose log fold change between cluster 2 and the rest of cells is at least 0.1. So what is log fold change? What you did is that you take the average of a gene in cluster 2. You take the average of the gene in other cells. You take a ratio of the two averages, then you take the log of the ratio. So the log of the ratio could be positive if the ratio is bigger than 1. It could be negative if the ratio is less than 1. But what you need here is that 0.01 is the what? It's basically you want it to be positive so that the genes are highly expressed in cluster 2, right? Rather than highly expressed in other cells. So you look for the highly expressed genes in cluster 2. And the second argument is the test.use. Here, the default is the Wilcoxon rank sum test, which we will discuss later. And just for your information, Sura has other tests for you to use, including negative binomial, Poisson, or DE-seq2. So these methods are all designed for count data. That's why if you use these methods, the slot, which is actually default set to data, which means normalized data, it was from the previous normalization step, will be changed to counts. Counts are the original data before you do the normalization. So you see Surratt default has this default change. If you use the count-based test, the data will be counts. If you use the default Wilcoxon, data will be the normalized data. That's something good to know, important to know. And there's another parameter, min.pct. It's the minimum percentage. That is, you want the gene you look at to be expressed in at least 1% of cells in both your group 1 and the rest of the cells, group 2. Otherwise, if the gene is not expressed, you don't think it's worth examining. Okay, so you're looking at one gene at a time. So this gene, it passed this log fold change threshold, it passed the min percentage threshold. So it was in the pool of examination. And then Wilcoxon is wrong on this gene, which we'll talk about later, and then you get output. Now let's look at output. What is the average log fold change? It is the log fold change of the average expression between the two groups. Positive values indicate that the gene is more highly expressed in the first group. All right, this is what we expect to have. So again, it's log 2 applied to the ratio of the gene's average in group 1 over the gene's average in group 2. And PCT.1 is the percentage of cells where the gene is detected, with a non-zero expression. So that's the percentage. PCT2 is the percentage for group 2. So you see that for these genes, right, which are found as marker genes for group 1, they actually have much higher percentages of expression in group 1 than in group 2, which is expected. And for each gene, Wilcoxon will give a p-value, which we will talk about later. So just as a precaution, what I found over the years is that very few people understand the p-values correctly. So which we will talk about later. Hopefully after today's lecture, at least our class, our students understand what is p-value, right? Okay, so let's bear this question in mind. And then in the last column, you see p-value is adjusted. It's called adjusted p-value. It's said to be based on Bonferroni correction using all genes in the dataset. What does this mean? Why do we need to do adjustment? That's another topic we'll talk today, the multiple test correction. Okay, so if you compare the two columns, p-values and p-values adjusted, you will see that p-values adjusted are bigger. In general, they're like at least 10 to the fourth, right? Around 10 to the fourth bigger, right? What does that mean? 10,000 bigger, 10,000-fold bigger. So basically, adjustment will inflate the p-values, so you will have fewer significant genes than if you just use p-values. That's the intuition. Later, we'll give you the detail. Okay, so second example, still in Surratt. Now our first group is cluster 5. Second group is not all cells, but the cells pulled from cluster 0 and cluster 3. So we just make the group 2 more specific. And we find the marker genes and we report the top 5. So you see that these genes look super significant, right? P-values are very small, 10 to the negative 200 something. After the adjustment, they remain to be very small, but bigger, yeah. So same result for interpretation. Finally, if you run find all markers, then Surratt will by default give you the marker genes for each cluster. So this is just an R operation to help you get a nicer structure. In particular, this dplyr is a very nice function, nice package written by the developer, Hadley Wickham, who developed the RStudio. And it's a very nice package for working with data structure. Essentially, using the filter function and using the pipe, you can directly go one step to another. And this one shows I'm only interested in the genes whose average log2 fold change is bigger than 1. All right. So you see that this is what we get. All right. So you still have the p-value, average log two-fold change, PCT1, PCT2, p-value adjusted. And for the clusters, you want to show which cluster you are looking at. And there are some nice visualization building in Seurat. I think this explains why Seurat has been so popular as a package, as a tool. So the violin plot shows every gene's expression distribution within a cluster. So this gene is found as a cluster 3 marker. So we're showing that indeed, looking at the violin plot, you can confirm the gene is much more highly expressed in cluster 3 than in other clusters. And same for this gene. So violin plot, basically to interpret this, you should look at it vertically. So basically, if you rotate the graph by 90 degrees, the curve on each side shows the density, right? So this is where the density is highest. It's about this expression level 3.7 or something. So that's how you interpret the violin. It's actually a modified plot of the box plot. In the box plot, you just show the median, the 75th quantile, and 25th percentile, I should say. And also, I think probably 90 or 10 percentiles, and then outliers. You just show those important critical values, quantiles in your data. But violin shows the density. All right, another example showing two more genes. So you can see that their distributions are very different between different clusters. Besides, it has this function called, oh, by the way, the function is called VLN plot, standing for violin plot. And you also have the feature plot. Feature plot, what it does is that to show a gene in the UMAP plot. So the cells are positioned as in a UMAP plot, and the color of the cell indicates gene's expression value. So the darker the color, the more highly expressed the gene is. So therefore, you can see, oh, this gene is highly expressed in this cluster, and this gene is highly expressed over here and here. So you can see that. And furthermore, you have heat maps. Heat map is also a very useful way to see the data. Here you have the rows as genes and the columns as cells, where the cells are grouped by their cluster label. And clearly, what it does is to group the genes. So it's only showing, I should say, it's only showing the marker genes. Maybe the marker genes returned by find all markers. And it's only showing the top 10 marker genes for each cluster. So you have a better visualization. And clearly you see these genes are highly expressed in cluster 0. These are high expressed in cluster 1, cluster 2, cluster 3, cluster 4, 5, 6. But interesting thing is that for cluster 6, and it has a very big overlap with another, how to say. So these genes, I should say, the genes highly expressed in cluster 4 are also tend to be highly expressed in cluster 6. Right? This is some information you might use. Because, for example, you might want to have a question, oh, should cluster 4 and cluster 6 be merged? Because if they have similar marker genes? That's just one way to think about your results. Okay, then in Seurat, it assigns cell type identity to clusters based on the genes. So the thing here is that All right, sorry, the internet was disconnected. So let me redo the screen sharing. This is where we were at. So I was saying that when you look at this marker gene CD8, it tells us, oh, this cluster is likely CD8 T cells. So we use knowledge to help us do the matching. And that's still the most common way for annotating cell types. But you can see the challenge is that when you have some clusters with genes you're unfamiliar with, then maybe that's the chance for finding or annotating a new cell type. But sometimes it's really depending on the people who do the analysis, right? It's subjective. You can keep it as a so-called new cell type, or you may merge it into some existing other clusters. So therefore, there's also a discussion or lack of consensus on what cells should be called cell types or versus cell subtypes. And also some people like to use the term cell states, which are considered to be kind of continuous. If you have some cells that appear to be between two cell types, and it's hard to say which one it belongs to absolutely, then some people say it's a cell state. So I think with the single cell technology and the data available, the definition of cell types are being, how to say, updated. And for many papers, the consensus is still not there yet. So that's why there is an international effort to build a so-called human cell atlas that aims to have a cell atlas for every human tissue and organ so that we can use the annotation there as the reference to provide a unified annotation system for other data sets, for new data sets. All right, so now I'll talk more about the statistics underlying. So what we are actually doing to find the marker genes in the D step, we are essentially doing the so-called hypothesis test. So this is something I think everyone should have learned in an intro to STAT course, but I want to re-emphasize what it actually means because it's not just a procedure. In fact, it's a way for us to think about our data and how to connect how to use our data to help us decide whether our scientific hypothesis is reasonable or not. So the first step is to list our null and alternative hypothesis. The null hypothesis is a statement of no effect, no difference, or no change. It is the hypothesis that a researcher seeks to test and potentially reject. Typically, it's the thing that we treat as the status quo or the default, and we want to use our data to tell us whether we have a strong enough evidence to go away from this default and reject it. For example, H0 might state that the mean of a population is equal to a specified value. So in our case, when we compare a gene, to decide whether it's a marker gene of a cell cluster, what we're doing is to decide, okay, does the gene have the same mean in this cluster of interest and the rest of the cells? If that's the case, the gene is not a marker gene. Okay, so basically null hypothesis in our case is the gene is not a marker gene. And alternative is the opposite. It's a statement that indicates the presence of, in fact, a difference or change. If it is what the researcher wants to prove, the alternative hypothesis is accepted if the null hypothesis is rejected. For example, we offer H1 as opposed to H0 for the null. H1 might state that the mean of a population is not equal to a specified value. In our case, the alternative hypothesis is that the gene has a higher expression in the cluster of interest than in other cells. Second, test statistic. The test statistic is what we construct to summarize our data. So as we will see later for the two sample t-test, we summarize our data into one number, and that number is a test statistic. We hope that this number is informative. It can tell us whether the null hypothesis is reasonable or not. That's the intuition we have. And just for information, even though in the intro to STAT class, you only learn the standard statistics such as z-score, t-statistic, chi-square statistic, and f-statistic. I don't know if you learn all of them, but at least you must have heard about z-score, t-statistic. But I have to say they are not the only ones you can use. You can invent whatever test statistics that are reasonable. And those traditional ones, they became traditional because in the old days, we didn't have enough computing power to derive the third step, null distribution. So that's why the statisticians 100 years ago, in the lack of computers or even calculators at that time, they help to provide some standard null distribution. So you can look up a table in the book, right? So printing those tables is like a big task back then. Then with the tables, you use a same table, same null distribution. You only want to know the value of your z-score or your t-statistic. Then you compare your value to that table to make a decision. It's just for the convenience at that time. But now I want to say that we have computers to help us get a null distribution. Then we are no longer restricted to use the standard or traditional or conventional statistics if the problem is not standard. So that's just something I want to convey here. All right, so the test statistic is a standardized value that is calculated from sample data during a hypothesis test. It is used to determine whether to reject a null hypothesis. The choice of test statistic depends on the type of data and the specific hypothesis being tested. So therefore, I just want to say that whenever you summarize your data as a test statistic, you can always derive the third step, null distribution for the test statistic under the null hypothesis. But some test statistics may be more powerful than others. By being more powerful, intuitively, what it means is that the test statistics distribution under the alternative hypothesis is more different from its distribution under the null hypothesis. So the bigger the two distributions are for the test statistic, the more powerful it is. That's the intuition, because you want the test statistic to help you separate the two hypotheses. But if the test statistic in the extreme scenario follows the same distribution under both hypotheses, then it has no power to do the distinction, and it's not so useful. All right. So why do we have a distribution? This is something fundamental in statistics. Here, the distribution is what we have in mind. It's not something we observe. So what we're thinking is, oh, if the null hypothesis is true, under these assumptions and logic, then my test statistic should follow this distribution, which means that the test statistic is not taking a single value. It can take multiple or even infinitely possible values under the null hypothesis. So given that, it's kind of a thought experiment. Given we have this in mind, then we evaluate whether the value we observe from our data here can be considered as too extreme, or it seems to be very probable given the null distribution. That's the underlying logic. And I think the challenge of statistics is also in this null distribution. We are talking about things we don't see. It's actually in our mind. And I don't know how many intro to stat courses emphasize this concept, but I highly doubt it, because I think many people follow the procedure, like how to do t-test becomes just a procedure, right? You do the calculation, then you convert the t statistic to a p-value, then you compare p-value to 0.05, for example. It's like a procedure. And this step is kind of underlying. But what I want to emphasize here is that this way of thinking about your statistic to have a distribution is what we call frequentist statistic. What it means is that we believe the data we observe is just one of the many possibilities. And when we talk about whether we believe the null hypothesis to be reasonable or not, to reject it or not, we are actually thinking beyond our data. We're not just using our data as the only source. We have the assumption underlying to help us decide whether we have enough evidence. Because it's possible that, okay, if you calculate that log4 change is not equal to 0, right? Because 0 means that the two averages are equal to each other. But it could be different from 0, say 0.01. But do you think 0.01 is big enough to let you reject the null? That's the question we want to answer using the null distribution. Because whether 0.01 is big enough depends on what the null distribution is. If the null distribution is very concentrated around 0, then 0.01 may be significant enough. But if this is very wide, then 0.01 is not significant enough. All right, so the null distribution is the probability distribution of the test statistic, assuming that the null hypothesis is true. This distribution is used to determine the likelihood of observing the test statistic under the null hypothesis. The shape and parameter of the null distribution depend on the test being used. Say normal distribution, the standard normal distribution for Z tests, T distribution for T tests. And these standard distributions, like I said earlier, just now, were constructed for convenience. So we don't need to work with so many different distributions, just the standard ones that can be made into tables. Finally, convert your test statistic to a p-value and then make a decision. So here, what is a p-value? The p-value is the probability of obtaining a test statistic at least as extreme as the one observed, given that the null hypothesis is true. It quantifies the evidence against the null hypothesis. So it seems still quite abstract the way it's said, right? So p-value, in short, is just a conversion of your observed test statistic value. So given the null distribution, you put in your value, and then you evaluate what we call as a tail probability of the value. Basically, what is the chance that you observe values more extreme than your current value? So that depends on what you call as extreme. Because if you think extreme just means larger, then your p-value is just the right tail. If you think the extreme be more extreme means that larger in absolute value, then your p-value will be the two tail probabilities sum, right? Left tail plus right tail. So it depends on what you consider as more extreme. But p-value is just a conversion of your test statistic. So given every test statistic value, you can get a corresponding p-value. Okay, and that's just another summary of your data. So p-value is also a summary of your data. So if you believe that the null distribution is true, and then if you believe that your data, so basically if the data does come from the null, then the null distribution should be accurate. Then your test statistic should exactly follow the null distribution, right? So for every value your test statistics get, it's a sample drawn from the null distribution. Then if you apply the p-value conversion, the p-value as a tail probability should be what? It should follow uniform 0 to 1 distribution. So that's why we say, if you believe the data is random, a random draw from some distribution, then the test statistic value is random. Then the corresponding p-value is random. What you observe is just one of the many possibilities. All right, so we're understanding this p-value randomness. I think it's not something most people have. You may think p-value just as a value between 0 and 1. A lot of the common misuses of p-value is due to this misunderstanding. So the key thing is that you must have a valid null distribution of your test statistic, so you can get a valid p-value. So some common example of misuses in bioinformatics include that. I can give you one example I recently saw. That is, in our differential or in our gene example, you get one p-value you get one test statistic per gene. And what the paper does is that it pulls all genes' test statistic values together, call that a null distribution, and use that as a null distribution to give every gene a p-value. What's the problem with that?
So this guy takes a p-value for each gene and aggregates it.
No, it's not. So they first aggregate. So every gene has a test statistic value. It pulls the test statistic values across genes to form a null distribution. Call that a null distribution. And then use that null distribution to convert every gene's test statistic value to a p-value.
So
you're confused by my statement or why they do this?
So they make a null distribution using the test statistics.
Yes.
And then use that same test statistics.
Yes.
To convert it into this p-value using the null distribution just from the.
Yes. Right. So in this case, the p-value is basically ranking what they did because it's just the percentile of the gene among all genes. That's it. P-value provides nothing beyond this ranking. Yeah, so your intuition is right because they didn't do the background. Yeah, so you know what is fundamentally wrong in this approach is that by pooling all genes test statistic values to form a null distribution, there is an assumption. The assumption is that all genes are null. Yeah, if you believe all genes are null, what are you discovering? Right. So I'm not proposing. I'm putting out this example because I was very shocked. And I can say that this was published in a high impact journal. Yes. So yeah, so that's why, you know, and also when I talked to some of the bioinformatics people, they told me this practice is not so rare. You have?
No, I have never seen it.
Yeah, I think it's possible that it's not so common in genetics because I think maybe genetics people use statistics better.
Okay.
Yeah. Yeah. But anyway, so, okay, I can say that this is something you might be able to see, especially if p-value is not the focus of the paper. I have to say in this case, the focus of the paper is the test statistic they propose. Okay, there is some rationale or intuition underlying the test statistic, but the p-value is wrong. So if the p-value is wrong, what does it matter? It will make you not able to say anything about your threshold. Where do you threshold the p-value? Right? If it's not a valid p-value, then you can threshold it anywhere. Okay, so just to maybe elaborate a little more on that example, the problem is if you use 0.05 to reject the genes, right? Using p-values under 0.05 to reject the genes. In that approach, you will always reject 5% of your genes, no matter what, right? But that shouldn't be the case because there is possible that your genes have nothing important, or it could have more than 5% of genes of interest. But using that approach, you will only reject 5%. Anyway, so once you have the p-value calculated, you can compare the p-value to your predetermined significance level, which is called alpha. And it's usually said to be 5%. But I have to say this 5% is arbitrary, right? This 5% was just because Ron Fisher, who just said, oh, 5% seems to be a small number. Yeah, but it could be 1%. So, but this alpha is telling you, even if your genes or your data are all from the null, there is 5% that you rejected. So it's the anticipated so-called type 1 error. Okay, now two sample t-test. This is what everybody learns in, again, Intro to Stat course. So here, what we're trying to compare is that you have two samples. So this is a very interesting example I found online. It's about whether we want to see the pirates who wear eye patches have fewer tattoos on average than those who don't wear eye patches. So we divide the parrots into two groups based on eye patches, and we compare their tattoos. So we have a distribution of tattoos in each sample, each group. And our null hypothesis is that the two parrot populations have the same mean in the number of tattoos. From the data, we get one sample average for the eye patch group, 0.34. Another average for the no eye patch group, 9.61, right? Seem to be not so different, right? And we're showing the data distribution. You can see the distribution is relatively wide. So because of that, then this subtle difference, like 0.3 difference, might not mean something. That's our intuition. And we can formally do this by a t-test. So in R, the t-test can be done in two ways, using the regression formula or by comparing just two vectors. So the null hypothesis is that the two populations, not the two samples, the two populations where the two samples are respectively from have the same mean. And this is the t-statistic. You take the average of group 1, sample 1, minus average of sample 2. In the denominator, it's for standardization to divide the standard deviation of x1 bar minus x2 bar so that you can use the standard t distribution as a null distribution. That's it. Because otherwise, if you don't do the division, the difference along the distribution, the null distribution of this x1 bar minus x2 bar should have something to do with their standard deviation, right? So then you cannot use a standard t table. That's the reason. There are several requirements for the two-sample t-test. First, the two samples must be independent of each other. That means the data collected from one group should not influence the data collected from the other group. Second, the data in each group should be approximately normally distributed. So this example actually shows that it's kind of like a bell shape. If the sample size is large, like if you have more than 30 data points per group, per sample, it's not so much of a concern. The reason is that we are working with x1 bar minus x2 bar, two averages, and we have the central limit theorem. Central limit theorem is saying that, okay, when we take the average, as the sample size goes large, it's an average of more data points, it will more behave like a normal distribution, the average itself. So therefore, if x1 bar is normal, it's approximately normal, x2 bar is approximately normal, then their difference is approximately normal. And that satisfies the requirement. That's something I hope everybody bear in mind. So that's why two-sample t-test is beyond that testing two normal samples. Okay, third, in the denominator formula, we're pooling the standard deviation square of sample 1 and standard deviation square of sample 2. We normalize each one by the sample size and we add them up. So this idea is assuming the variances of the two populations are equal. This is the equal variance two-sample t-test. If this assumption is not reasonable, we should use the Welch's t-test as instead. Okay, so but this is good when N1 and N2 are small, right? Because when they're small, using one sample along, say 3 data points, if N1 is 1, to estimate the variance is not accurate. Then you better pull them together. Again, if the sample sizes are small, it's hard to say whether you believe the two samples have equal variance or not, right? So it's better to be simpler. So therefore, I think the more assumptions are associated with smaller sample size. That's typically the logic. If you don't have enough data, you have to make more assumptions to gain some power in your analysis, but you have to justify that. This is also showing the subjectivity in statistical analysis. Okay, Wilcoxon rank-sum test, the default using Surratt, is also run on one per gene. So it is also known as the Mann-Whitney U-test. It is a non-parametric statistical test used to determine whether there is a significant difference between the distributions of two independent samples, same as two-sample t-test. But it does not have the parametric normality assumptions So it doesn't need to assume this is X1 bar is approximately normal, nor X2 bar is approximately normal. It has the several features. It is non-parametric. It does not assume that data follows a specific distribution. It uses ranks instead of raw data. The test converts the data into ranks and then compares the sums of these ranks between the two groups. This ranking helps reduce the impact of outliers and skew data. So basically, if you have N1 data points in group 1, you have N2 data points in group 2, you will pull them together and rank. Then you will see whether group 1 has a larger sum of ranks than group 2. Because if the two distributions are actually under a null hypothesis that the distributions of both groups are identical, if you believe your two samples are from the same distribution, then the rank sum for group 1 or sample 1 and the rank sum for sample 2 should be similar, right? So you shouldn't have larger ranks on either side. All right, so basically this is the test statistic. R is the sum of the ranks for the smaller sample. You take the smaller rank sum and you do the standardization to convert it to a z-score. So you can use z distribution or standard normal distribution as the null distribution for convenience. But these two values, the expectation of R under the null and the standard deviation of R under the null, these were theoretically derived. All right. So then you can use the z-score as the test statistic. But like I said, these are for the mathematical complexity was used for computational convenience. So you just use standard normal. But what if you don't have the capacity to derive, oh, this is the mean under the null, this is standard deviation under the null. What do? You can now use computer. You can do permutation to provide the negative samples, right? Then you can use that to give you a null distribution. So permutation is kind of a universal strategy to get a null distribution with test statistic, but it is computationally intensive. All right. So what is multiple testing correction? So in our example, we do one test per gene. So the number of tests is equal to number of genes, but there is a problem of multiple comparisons. When we perform a single hypothesis test, the probability of making a type 1 error, false positive, is equal to the significance level, commonly set to 0.05. So that is what I wrote here. So the probability of having a false positive for one test is 0.05. Then 1 minus that is the probability of having no false positive for one test, 0.95. This means that there is a 5% chance of incorrectly rejecting the null hypothesis when it is true. However, when conducting multiple tests, the probability of making at least one type of error increases with the number of tests. So for example, if you perform 20 independent tests, each with an alpha of 0.05, the probability of making at least one type of error is no longer 0.05, but can be 0.64. It gets larger as you do more tests. The reason is by this probability calculation. So the probability of making at least one false positive is 1 minus the probability of making no false positive in the 20 tests. So as the 20 tests are independent, it's 1 minus that each test having no false positive to the power of 20. So it's like 0.95 multiplied to 0.95 and so on for 20 times. And you can see this second number will decrease as 20 goes up. So because the bigger the power, the smaller the number. So that's why we get 0.64. And you can see that we will more likely have at least one false positive if we do more tests, even by random chance. That's equivalent to true sample t-test. Yeah, regression actually has this equivalence between the two. And regression is regression can account for more factors. Like here, we only have a group factor, right? 2 groups. But if you have some confounders to adjust, you can do a regression. Yeah. Yeah.
So we mentioned this permutation. One question that I always have in mind is, let's say you don't have enough computational power. You only did, let's say, 10,000. Maybe go up to that, you can go down to
Yeah.
And, you know, it takes the idea of, you know, you look at this small distribution, 10,000. It's a distribution on top of this.
That's common. That's actually common in practice. You use permutation to give you a coarse distribution, and then you use the parametric distribution to increase the smoothness, basically. Yeah, exactly. Actually, I wouldn't call it estimate. I would say the permutation give you an so-called empirical null distribution. And you want to make the empirical null distribution, which is the discrete distribution by nature, want to turn that into a continuous distribution. But you know what's very important is sometimes you don't know there is a theoretical distribution underlying, but there might be one. So once you use permutation to get you this empirical distribution, if it fits extremely well to a Gaussian distribution, for example, or a gamma distribution, then you might doubt maybe there is a theoretical distribution underlying. Yeah. Yes. I would say at least 100.
Yeah.
Yeah. And I'm sorry, so basically it really depends on your sample size, because if if your sample size is very small, you cannot afford to do many permutations because you might be exhausting all the possibilities and you're getting repeats. Right? So basically, if you're saying, okay, I have N1 plus N2 samples, and I want to randomly allocate them to two groups and keep the sample size, the biggest number of permutations you can afford is N1 plus N2 choose N1. Yeah, so that's why permutation wouldn't work well if your sample sizes are too small. Yeah, because that's just not enough possibilities. Yeah, and also you can Google this term called balance permutation. So for balance permutation, what it tries to do is that it was to try to maintain some balance of the original two groups so that you will not use the chance where you just put all the original N1 into one group, original N2 into one group. Because even though that comes from permutation, but it's just the same as your original data. Yeah, it's not a reasonable background. Okay. Thank you. So this is the multiple test correction need, because otherwise we might have more and more false positives as we do more tests. So there is a quantity to measure this. It's called family-wise error rate, which is the probability we already saw in the last slide. It's the probability of making at least one false positive. So besides this probability that will go up as n goes up, n here is the number of tests or number of genes. We also expect the number of false discoveries to increase, because if your m tests are independent, right, each test has alpha to be a false positive, then expected number is m times alpha. And this number will also go up as you have more tests. To actually control this family-wise error rate, instead of controlling just the type 1 error for one test, we have this so-called Bonferroni correction, which is what's used in Surratt. Bonferroni correction is very simple. It just adjusts the significance level by dividing alpha by the number of tests. For example, if alpha is 0.05 and there are 20 tests, then your adjusted alpha is 0.05 divided by 20. That becomes 0.0025. It's simple. This Bonferroni correction is simple and conservative, but can be overly stringent. So you may not have any p-values below this threshold. that means you have no discoveries. That's possible. So you can see what we're doing is we compare every gene's p-value to this 0.05 divided by number of genes. And we can do it the other way. We keep 0.05, but we adjust the p-value. That means we just multiply p-value by n and then compare that adjusted p-value to 0.05. And that's what's done in Seurat. The p-values are multiplied by the number of genes. That's why you can see the p-values after adjustment are scaled by about 10 to the 4th. That's the number of genes. But for your information, this approach, just remember how small the p-values we get out of Seurat. Remember that 10 to the negative 200. That's for a reason, which I hope I can cover in the end. But anyway, because Bonferroni correction can be super stringent, making you have no power, in 1995, there was a phenomenal paper by Benjamin Hochberg that proposed this idea called FDR. So FDR is proposed as an alternative criterion, as an alternative to a family-wise error rate. It's a different definition. So it is used, it's measure multiple tests to control the expected proportion of incorrect rejections of the null hypothesis. It is the expected proportion of discoveries that are false, right, among all discoveries. So basically, the formula is that you are taking the expectation of a ratio, number of false discoveries over number of discoveries. And to avoid the dividing zero, you have the denominator to be at least 1. Here, the expectation is taken over the distribution from which the data is sampled. So for your information, this ratio in the expectation is never observable because we don't know on our data what are the false discoveries. We only know how many discoveries. But the interesting of statistics is that even though you don't know this ratio, but you can control expectation within the Benjamin Hochberg procedure. All right, the two criteria are different perspectives. So basically, controlling the FDR is less difficult than controlling the family-wise error rate. So therefore, if you're not so conservative, you just want to control the FDR, you might still have discoveries. Okay, so they have different ways to set p-value thresholds. Let's read this. The family-wise error rate controls the probability of making at least one type 1 error among all tests, which can be conservative. The FDR, on the other hand, is less stringent and allows for a proportion of false discoveries, making it more powerful and suitable for large-scale tests, especially when M is big. What is the Benjamin Hochberg? So it's based on ranking of p-values. So it will compare the ranked p-values or order p-values to an increasing threshold. So specifically what it does is that you compare the k smallest p-value to this threshold, 0.05 divided by m multiplied by k. K is the order. So as k increases, this threshold increases. You continue until you have a p-value that exceeds the threshold. Okay, when you stop, then you reject all the previous p-values that satisfy that p-values under the threshold. This is a cartoon illustration. So y-axis is the order p-values from smallest to largest. The straight line marks, okay, what is the threshold? It changes as your k increases because it's alpha divided by m multiplied by k. So k is the slope. And then you compare each p-value to the line. You stop until the p-value goes above the line. And then you reject all the p-values that were consecutively below the line. That's your rejection. So you see that compared to Bonferroni, the threshold you compare to is bigger. So you are more likely to have rejections. All right, but what I want to say here is the FDR control requires valid p-values. By valid, I mean that the p-values, if they are under the null hypothesis, they should be distributed uniformly between 0 and 1 if the test statistics come from the null. Otherwise, it will indicate that you don't have valid p-values. A way to check it is to plot the p-values across your tests to see the shape. Because we should expect to have a relatively uniform distribution for most of p-values, but some small spike near 0 to indicate these are the things we want to reject. But if you have a histogram like this, a very sharp spike just below 0.05, this might indicate p-hacking. That is, these p-values were selected, right? You look at your results and you screen for the smallest p-values to keep them. That's the problem. I think this is in the article I shared with you after the first lecture. Okay, so what is p-hacking? For example, you do multiple tests, say 20 tests, you only report the 3 tests that are significant below 0.05, and you ignore the rest. So then this is not right, because you may have small p-values even though all the 20 tests are null, right? You can have a small p-value just by chance. But if you ignore the 17 non-significant p-values and only report the 3 significant p-values, you are doing p-hacking. All right. So that's all I want to say for the multiple testing. I know we are running out of time, but for the remaining, I will just quickly say that for single cell data, the reason why Seurat gave you such small p-values is because of the so-called double dipping issue. So this is something my lab studied on. What it means is that the genes use the same data to divide cells into clusters. Then you do the DE test between clusters. So therefore, if 2 genes are involved in clustering, they will naturally become DE genes. But it doesn't mean that they are marker genes, because both genes could have very continuous distribution across the cells. It doesn't mean that they are a marker of one-celled cluster. So I'm just showing that. So using this default approach in Seurat, which I call naive or double dipping, you will have the p-values not uniform. Even though all genes are non-marker genes, of course, this is from a simulation. If all genes are non-marker genes, you will still have p-values concentrated around 0, even though it should be uniform. So this is a problem that p-values are not valid due to double dipping. So something my lab proposed as a fix is that we hope to generate cells that are in one homogeneous cell type in our model, using our modeling to serve as a negative control. So we know that these cells we simulate shouldn't be divided into cell types or cell clusters because they are like one type. Then it serves as a negative control for our discovery. All right, so basically we added this synthetic null data generation on top of the Seurat pipeline. So you can run Seurat on your real data and the synthetic null data in parallel. And you compare every gene's DE results from 2 data sets to make discoveries. So due to the time, I will quickly jump to this page. So basically, my group 3 years ago, we adopted a statistical recent events to propose p-value free FDR control. So basically, if p-values are difficult to obtain and the p-values you get are not valid, you don't know how to get valid p-values, you can use this computation-based approach instead of using p-values to control the FDR. So without going into the details, because the slides are online, I'll just quickly show you one example to see that what we change. So here, the x-axis on the left are just the negative log Seurat p-values. You can see the p-values are very big. P-values are very small, making the scores very big. So like this is 10 to the negative 30. This is 10 to the negative 35. So what I'm seeing here is that on a data set where we know there are 2 CD14, CD16 cell types, monocyte cell types. So these marker genes and the housekeeping genes, they shouldn't have similar p-values because housekeeping genes are highly expressed everywhere. However, some housekeeping genes by Seurat can still have very small p-values, right? There's some overlap between the 2 sets on the x-axis. Using our approach, we have the null data DE scores, and then we do a difference as a contrast score. Then we see by the contrast, the 2 sets are better separated, meaning that if the housekeeping genes are here, they also receive high null DE scores. So therefore, after cancellation or computation, the housekeeping genes do not have as big contrast score as the marker genes. So we can better select the marker genes on the top. So that's just something we have in mind to kind of, I would say, to avoid this double dipping issue. But I think I didn't have time to talk about how we do this p-value free FDR control in detail, but I think I should just mention that there's something out there. So you don't have to really use p-values as the only approach if you want to look for discoveries. All right, so that's all I want to say. And as I mentioned that for next Monday, it will be just an office hour. And well, I'll be on Zoom to answer your questions about the final project. And the final presentation will be in the morning of June the 14th. I think it's a Friday. So you still have plenty of time, right? More than 2 weeks to prepare that 5-minute presentation. And you will have the project due by June 16th. Okay.
Sorry. So no in-person class next week,
just a Zoom office hour. Yeah, no in-person class next week.
And then Wednesday. So Monday you have, but Wednesday there's no class.
Usually, I don't think we don't need. So basically, I don't have plan to have the Wednesday for the office hour. But if you need more questions, I will ask Zhiqian, the special reader, to be on Zoom on Wednesday. Yeah. And also, your questions can be asked not just within the office hour. Yeah, you can also post your question to the website so that we can have everybody on the same page if it's about the final project. Yeah. I'm sorry? Ruler, yeah. All right. Any questions online? If not, then bye, everyone. Bye.
Bye.
Bye.